everybody, and welcome to our webinar, another webinar for our year. And this time I'm joined by my long-term friend, Dr. Christoph Krafft from the Leibniz Institute of Photonic Technology, otherwise known as IPHD, in Jena in Germany. If anyone hasn't listened to me before, I'm Dr. Mustafa Kansas. I'm the Director of Product Management at Photothermal Spectroscopy Corp. I've been doing these webinars for a long time, but this one is especially exciting for me because it brings together so many key and enabling capabilities. And today we'll really be talking about fluorescence guided submicron IR of white blood cells in blood smears on glass slides. And a few key words there, they are really that we're doing fluorescence guided. We're looking at specific types of cells in blood smears, and it's on glass slides. So I'm going to be stressing the glass component a fair bit throughout this talk. Like all of these webinars, a few little notes to keep in mind. This is live, so of course you can ask questions in the chat box. So please do. We always encourage those, and depending on how much time we have at the end of it, we'll endeavour to answer as many of those as possible. Anything that we don't, of course, we'll get back to you via email. And then really getting right into it, this particular webinar is going to be a little bit different in that I've added some extra background information for those of you who may be new to spectroscopy. I know that these webinars often have a lot of new attendees come along. And so the first couple of slides, I'll give you some background as to why one might do infrared and Raman spectroscopy for the life sciences. And then I'll do the sort of usual background on the existing technologies, the pros and cons. I'll then introduce to you how O-PTIR works and how it overcomes virtually all of those existing challenges. I'll take you through a few existing applications, drawing particularly and mostly from the published literature, and then I'll hand over for Christoph roughly about the halfway point for him to delve a little deeper into his recent research. So, infrared spectroscopy, and what does it bring to the life science research? Why would one do vibrational spectroscopy infrared? Well, infrared spectroscopy gives us the ability to do label-free spatial distribution of the key biochemistry. So we're talking the macromolecules, we're talking the proteins, lipids, nucleic acids, carbohydrates, and a few other components as well. So again, if anyone's not familiar with what an infrared spectrum of a typical biological component, a molecule or sample would look like, here we have absorbance as a function of wavelength or energy, and we've got a number of different bands, and these peaks are all related to the overall chemistry that is being probed at that point in time. So as I mentioned, most of these bands and peaks are from the macromolecule, so proteins, so secondary structures, these works out quite well. Collagen, lipids have really nice unique bands and peaks. So you can look at lipid chain length, degrees of saturation, nucleic acids turn up around about here, so you could look at key nucleic acid features, and carbohydrates also have nice strong spectral features as well. And that really gives us some simultaneous relative concentration of all of these key bio components or macromolecules, and this is delivered spatially at that submicron, sub 500 nanometer level, and importantly, it's also very non-destructive. Further to the macromolecules that we probe with infrared spectroscopy, you can even label sometimes. I mean, I've said this is label-free, but you can actually label things or use stable isotopic labeling with deuterium, for example, or heavy carbon, heavy nitrogen, or even include ultra-small molecular infrared tags, or these moieties, azides, alkynes, nitriles, and the like, and they can be incorporated to improve and enhance the selectivity and specificity as those particular bands appear in the so-called silent region. Now, what does the infrared spectrum tell us? It's important to remember that when we look at that spectrum that I showed you before, that particular spectrum comes from about a 500 nanometer spot, and it reflects the total chemical makeup, right? And of course, biological materials, cells, tissues, and the like, they're not pure components. They're made up of many things. And with infrared spectroscopy, you're looking at the average of its total chemical composition. So for the average cell, If you come back to our macromolecules, your proteins are likely to make up in the order of 60%, lipids maybe 10%, nucleic acids 20%, carbohydrates maybe 10%. Of course, these ratios would differ. So these spectra that I show on the screen here are the pure component spectra. And then the resultant real-world sample spectrum is really just a weighted average of all of those. And that's where all of these different features come from. You can see the protein bands being the most abundant, dominate spectrum. But you can also see these nucleic acid bands over here. You've got some lipid bands over here, the C8 stretching. So it's important to, again, to remember that per point, you've got a total chemical spectrum that reflects a weighted average of the total chemistries. So hopefully with that little background primer there, we'll get back into looking at some of the more instrumental components and functionality components. So I'll be comparing this to traditional infrared spectroscopy. So when I say traditional, I'm referring to these direct IR, FTIR, and QCL techniques. So one of the main limitations there is spatial resolution, because we're dealing with long wavelengths. Infrared wavelengths are long. So that limits your spatial resolution. Sometimes sample prep, if you have to, the best quality spectra are with thin samples. Now with tissues and cells, they're already pretty thin and easy to prepare, so that's not so much of an issue. But if you can't cut the samples, then certainly that becomes a problem. For those sorts of issues, ATR to some degree mitigates that, but it's a contact technique, so that sharp crystal there, typically germanium, has to contact the sample and you end up indenting your sample. A lesser known issue that I always like to talk about, because I think it's an important issue, is so-called dispersive and scatter artifacts. So in an ideal spectrum, if that's a thin film of polymer there in black, and we measure that in transmission with the infrared, that's what a good spectrum would look like. A good spectrum is a flat baseline near zero with nice symmetric peaks. But it turns out that the sample shape has a huge impact on the spectrum regardless of the chemistry. So if you take the exact same material, now make that a bead and measure that in transmission, well, the spectrum is very different. And this effect, in fact, is very strongly coupled to size and shape and just overall morphology. And so that's an important thing to bear in mind. Since in the opening slide, I did stress the glass component. Glass is a very common substrate used in biology, but it's not a common substrate used in traditional IR, traditional FTIR and QCell. And that's because glass is quite opaque. So that's our infrared spectrum or wavelength range. Here is a spectrum, a single beam spectrum of a 1 millimeter glass slide. And you can see that from about 2000 onwards, you've got absolutely no infrared light penetrating through the glass, which means your key fingerprint region is effectively lost. But this cutoff point has some variability in terms of where you would define that to cutoff, and that depends on the thickness. So standard glass slides have a higher wave number cutoff. Cover slips, which are much thinner, let a little more light through, and you might be able to get down to around maybe 1600, but still most of the key fingerprint region is lost, which is a shame because glass is so cheap, so ubiquitous. Reflection of glass generates artifacts that are typically not used. So most people in the traditional FTIR QCL space are using infrared transparent substrates like calcium fluoride, barium fluoride, zinc selenide. However, these are far more expensive, and they can be, in fact, they are certainly much more fragile, and some of them even have some biocompatibility issues. Another key and relevant, life science relevant consideration and challenge with traditional IR is water compatibility. So unfortunately, water has a very, very strong OH bending vibration at 1640. So that's that one there, that band there. So there's a dip in the transmission of water in the infrared. However, that unfortunately, some stroke of luck, overlaps very much exactly with the main protein bands. So the protein bands are typically often obscured by this dip in transmission because water absorbs. And the more water you have, the less light you have coming through, which is unfortunate because water is life. That makes things much more difficult. Now, in the vibrational world, infrared is one half, Raman is the other half, and that's another commonly used technique. And one of its pros is Traditionally, it's better spatial resolution than traditional IR, but it is plagued with autofluorescence interferences, and I'll go through these quickly. It is also typically a low sensitivity technique, and so it can take much longer. You've got to do, for any imaging, you've got to do a full hyperspectral point mapping. Laser sensitivity, damage or toxicity to any living organisms can be an issue, and the spectra can depend on which wavelengths you use. Now, fluorescence microscopy has been around for many decades and is well established, but it does require labeling, right, which can be time consuming. It can disrupt the cell function, so it can change metabolism, limit multiplexing, bleaching, and phototoxicity still. And I've pretty bold at this last one here, that's limited chemical information. So whilst fluorescence is great for its ability to locate where a particular organelle is, for example, or some other cellular components, it lacks any broad chemical information, for example. It can't tell you secondary structural information. And that's where infrared comes into it. So that's kind of the cue here for me to, or the segue into optical photothermal infrared, O-PTIR. O-PTIR is a pump probe technique where we pump with an infrared laser and we probe with a short wavelength, typically green or a 785 laser. The ability to use that short wavelength laser gives us submicron spatial resolution, so it's kind of right. In fact, it's exactly the same as Raman in that sense, but it has still the richness of the infrared spectral region. We work primarily in reflection mode, which is the easy way to operate because you don't really worry so much about thickness. But we generate, even though we work in reflection mode, we generate FTIR transmission-like spectra without any of the traditional artifacts and distortions. It's non-contact and the spatial resolution is wavelength independent. Well, how does it work and how does it overcome these challenges? Well, it all starts with a pulsed tunable infrared laser. That's our quantum cascade laser. And that is focused through this black, or denoted here at least as a black objector. We focus that down onto the sample. Now, being an infrared laser, the focus spot isn't that tight. You can't focus infrared light too tightly. So let's say that's about 10 microns. Collinearly, we introduce our probe beam. Our probe beam is a green beam. So that green beam can be focused much more tightly, say about half a micron, so about 500 nanometers. And it's really that tighter focus that delivers the spatial resolution, that 20x improvement in spatial resolution. And this is the part that you have to sort of wrap your head around if you're coming from an FTIR background. So what we do now, so we have this tunable infrared laser that's tuning the range of the infrared spectrum. It's pulsed, and as we tune it from one end to the other, at certain points you have sample absorbance. It depends on the chemistry of the sample, and it depends on what wavelength you happen to be tuning past. But when the sample absorbs that light, you get a little bit of heating, a few degrees typically. Now that heating, because the laser is pulsed, that heating is also pulsed. It's heating, cooling, heating, cooling. So that whenever anything heats up, you have a slight change in refractive index, slight expansion. So that physical change also changes how the green light is reflected back. Right? So we're actually changing the reflectivity of the sample because it's heating up ever so slightly. Right? So we're actually monitoring synchronously the reflected green response while we are tuning the infrared laser and extracting out those modulations. And in doing so, you effectively create a pure absorbance spectrum, right? Devoid of any scattering artifacts, because if there isn't an absorbance, you won't get a signal. It can be the most scattering sample in the world, you still won't get a signal. A typical sweep is in the order of a second or even a little under a second. And also taking advantage of the QCL, you can park it at a particular frequency or wave number of interest and then do single frequency imaging, targeted chemical imaging, which is much faster than traditional imaging. Another key ability that's enabled through the O-PTIR framework is the ability to collect simultaneous IR and Raman. And I call this the holy grail of vibrational spectroscopy, because these have always been traditionally very different techniques on very different platforms, done sequentially if you had to measure it on the same sample. But now, really for the first time ever, we are doing infrared and Raman from the same spot at the same submicron resolution at the same time. Now let me just take you through this very simplified schematic here. That infrared laser, quantum cascade laser, the QCL, is shone through our microscope objective, focused down not so tightly because you can't. At the same time, we bring in our probe beam, as I just mentioned before. Now that is focused much more tightly. That's where that photothermal magic happens. The reflected green light is then sent back to a visible detector, and I'm stressing the word visible because it's room temperature. There are no traditional infrared detectors that often require cryogenic liquid nitrogen cooling. So that visible detector, from that visible detector, we extract out the infrared signal through the modulations. But because we are using a Raman grade green laser, you've also got Raman scattered photons. They're always there, whether you use them or not, whether you like or not, they're there. It's effectively a side effect. So we're taking advantage of the fact that there are Raman scattered photons there which are shifted in wavelength. So we use a dichroic filter here that separates out only the Raman shifted light, sends it off to a Raman spectrometer. All the other light goes off to the visible detector for infrared signal extraction. And really, hey presto, you've got simultaneous IR and Raman again from the same spot, same submicron resolution at the same time. So that really, for the first and only time, enables you to take full advantage of the complementarity. Because they're not on separate instruments, you're not having to struggle to find the exact same spot. So you can now be sure that your infrared and Raman spectrum is indeed from the same spot. And also confirmatory. Right? So the IR can confirm the Raman and vice versa. Spatial resolution is always a key factor. We're doing microscopy after all, so we typically care about small things. And this is really going to be the only time I show you any equation. This is a simple approximation for spatial resolution, the Rayleigh criterion, which really says it's a function of wavelength in microns as divided by the numerical aperture of your objective. And if you were to plot that out, you can see that for the traditional FTIRs and QCLs in the fingerprint region, the informative part of the spectrum, we've got a spatial resolution here and wave number here. It's relatively coarse. You're in that sort of 10-ish part range of spatial resolution, and that's because it's wavelength dependent, because you're probing with infrared wavelengths. But for O-PTIR, you're probing with a monochromatic laser source. So that lambda is fixed at 532. You have a high NA objective, and that means your spatial resolution theoretically is around the 400 nanometer mark, and it's flat across the spectrum, just the same really as Raman as well. And that's really where that 20 to 30x improvement comes from. Now, one of the newer features we introduced, well, probably a couple of years ago now, was the so-called counter-propagating. So some of what I just showed you there had the infrared in green coming from the top. The infrared can also be delivered from the bottom, thus freeing up the top objective to not be a Cassegrain, but to be a visible objective instead. And when you're able to use visible glass objectives, you have a lot more flexibility. You have a lot more options with respect to objective choice. If you can bring the infrared from the bottom, that of course requires a thin infrared transparent substrate, or you can even work with glass, as we've already touched on. But in that case, the sample needs to be upside down like that. You can also have access to all of the usual optical microscopy objective options for enhancing spatial resolution, oil immersion, water dipping. You can sandwich your samples in a flow cell capacity or configuration, then use a glass cover slip corrected objective as well. So loads of options available there. And one of those I just mentioned before was the oil immersion. Come back there. This is something that I collected a little while ago now. That's one of my favorite images. This is a cheek cell, I think, from memory. An image here only at the lipid band, 1740, shows incredible detail in that image. If we zoom into that, we see some of these features are really even a tad under 300 nanometers. And that's a spectrum from one of those points as well. So you do have that broad glass feature, which we'll talk about and Christoph will talk about. But the protein regions, the lipid regions are all very much clear. Glass being a bit of a theme here as well. So these are now standard glass slides. So we've had a tissue section that's mounted with the sample is flipped upside down. You're illuminating from beneath, exciting the tissue, probing it from the backside of the glass objective, so of the glass slide, and you're generating regular looking IR spectra on a glass slide. Now this of course could also be just flipped over, and you could measure this in co-propagating mode, where the green and the infrared come from the top. Right, and in this case there is no subtle glass background there, because this is a tissue thickness that's in the order of sort of 7 to 10 microns, and at that sort of thickness, glass doesn't become a contributing factor at all. Right. One of our beliefs and sort of strategic goals as a company is to make vibrational spectroscopy accessible and valuable to the life science user. So in order to help that and help facilitate that, we've tried to align our workflows to traditional life science workflows. And of course, that still requires some degree of fluorescence. So I spoke about the label-free capabilities, which is all great, but fluorescence is such a established technique, so widespread. And fluorescence certainly has some huge advantages when it comes to the specificity, for example. So we've really tried to bring the best of fluorescence and the best of infrared together by combining these two modalities into a single platform, single measurement. So you can now label your samples if you need to. You can highlight particular regions of interest and then go in there with the infrared spectroscopy to give you the chemical characterization that fluorescence sometimes lacks. So it's really the best of fluorescence specificity, and then coupling that with the best of infrared spectroscopy, which gives you that broad chemical profiling. So here are some labeled cells collected on the O-PTIR platform. They've been labeled for probably DAPI for the nucleus. There we've got the lipid droplets have been labeled in green, and a stress granule has been labeled in red. So so now the fluorescence image actually guides our spectroscopy. We can use this image to tell us where to collect spectroscopy, infrared spectra. So not surprisingly, a spectrum collected on the nucleus shows elevated nucleic acid features. Spectrum collected on a lipid droplet shows a whopping great lipid signature there. And then a spectrum collected on this stress granule here shows a change or a shift in the protein bands, potentially indicating some change in secondary structure. Now, when it comes to applications overview, so we are focusing on life science for this particular webinar, but I think it's worthwhile to mention that this is a tool that is used across all sorts of applications, well beyond life science as well, from polymeric applications to microplastics, particulates. We're doing a lot in pharma at the moment. Contamination in the electronics industry, semiconductor industry, it's also used heavily there as well. Chances are that your phone manufacturer is perhaps using one of these as well. But we are here today to talk about life sciences, and there it's been used across single cell analysis, bacteria, live cells, and I'll show you some of that. A huge area that I believe strongly on is neurodegenerative disease research, because a lot of that involves changes in secondary structure for which infrared is ideally suited. But from soft tissues to hard tissues, it all works really, really well. And one good measure of how well a technique is being adopted is publications. How are the publications rising over the years? And we started in 2008, and this strong exponential growth is very rewarding as a product manager for this technique. And I touched on the range of applications just before. When we dug into where people are publishing, it wasn't too surprising that it was life science was perhaps the most of these, but you can see a really good spread from life sciences to microplastics, a little bit surprised there with cultural heritage, and then the usual sort of polymers and material sciencey applications. And many people are also getting front covers. But coming back to, I suppose, the topic here, which was fluorescence guided, fluorescence co-located. The first publication with a researcher we work a lot with, Oksana Clementieva, came out in early 2023, where she used fluorescence to guide the infrared measurements, which is kind of what we wanted to do. And here's a nice example of where these were Alzheimer's disease mouse models that have been labeled with Amitreca 630 to highlight where the misfolded protein aggregates are. But we can image this in the bright field channel, in which case you see some contrast, but when you flip across and start imaging this with the fluorescence channel, you get a lot more contrast. And that's what fluorescence is all about. It's to help guide and locate where features are, where they are spatially. And once you can see where they are, that some of them you may not be able to see sometimes in the bright field, you can go in there with infrared spectroscopy. In this case, we drew a line profile. We took spectra on the hotspot, spectra off the hotspot. So you can see that the blue spectra from the hotspot had a very distinct beta sheet structure to them. Not surprisingly, these are misfolded protein aggregates, these plaques. Roxana Clementeva also published another paper in late 2023 where she looked at hydrated tissues. And this is something that actually I would not have thought was even possible, but she's done it. I'm glad she didn't ask me. She's taken freshly excised, fully hydrated tissue biopsies. Normally these tissues for traditional IR would be sliced, hydrated, fixed, and the like. But here, these are fresh tissues sitting in buffer, and then she's imaged them. So here's a protein band. You can see the protein secondary, so the beta sheet structure. Again, these are in water, and I'll be touching on how we are able to collect good quality protein spectra even in water. Right? So she's looked at living organisms, salamander embryos. She's measured the tails of these and looked at lipid droplets. I don't have room in this slide to show that, and these went on to live happy lives as well. Right. Caitlin Davis from Yale in the US, she's looked at de novo lipogenesis, lipid metabolism pathway, and she's looked to see the heterogeneity of that across cell types, right, all label free. And so we've got a bright field image, a single frequency lipid image, and then she's looked at, she's actually fed the organisms, these cells, carbon-13 labeled glucose. Here we are, labeled glucose. And then she's looked to see how that carbon-13 labeled glucose was metabolized into carbon-13 labeled lipid. So started off with C12 labeled lipid, but with the introduction of the C13, this added peak, which is shifted because it's now a heavier molecule, a heavier atom vibration. So the progression of that ratio between these two bands over time is a measure of rate of metabolism. And we can take advantage of the single frequency imaging here to image at the C12 band, at the C13 band, and then ratio those. So now we have the intensity of this color tells us something about the ratio of that band, of those two bands. So that's really where the spatio-temporal heterogeneity comes from. We can image these as a function of time. So you have the temporal and you have the spatial components there as well. Right? And this would all not be possible with a traditional FTIR because these are actually live in water, in nutrient buffer here as well. And with Raman, this would be, you could not do a single frequency imaging in Raman. Right, so the last two examples have all been hydrated or water-based measurements. So how is it that O-PTIR is able to measure in water when traditional traditional FTIR and QCLs can? And that's because with really O-PTIR, water, though still a feature, is much less of an interference. And that's because O-PTIR is actually an indirect measurement. So we're actually measuring changes in temperature. We're not actually measuring infrared photons as they go through the sample. We're using the infrared to excite the sample, generate a slight change in temperature, and then we're looking through the green beam at those changes in temperature. Right, so that means now it's not just the optical absorbances, heat capacity contributes to O-PTIR signal generation. Right, so for example, in this table here, I've got water listed for its heat capacity. So heat capacity is basically how much energy does it take to increase the temperature of a particular material. So water requires about 4.2 joules per gram per Kelvin temperature to increase it by 1 degree. Right, so 4.2. Now, if you compare that to connective tissue, so basically muscle is much less, and fat, which is adipose tissue, is mostly fat, and there also it's about 2.3. So there's a significant difference between water and other biological components, which means water doesn't heat up as much, so that the tissues and the fat will heat up more. Now, when I say more, we're talking still a few degrees. So that difference in its heating is actually helping to suppress the water background contribution, making O-PTIR much more viable for hydrated or liquid water sample measurements. Moving on to other examples, here's one from Roy Greater's group at University of Liverpool. They do a lot of work with bacteria, and I like this because it shows very clearly how the submicron resolution really helps pick out bacteria, single bacterial cells. Now, actually, I did my PhD looking at polyhydroxybutyrates with bacteria. Never in my wildest dreams could I have imagined getting individual bacterial images and spectra. So he's used 1656, the protein band, to locate where the bacteria are, and then a ratio of 1727 to that protein band, because 1727 is the polyhydroxybutyrate. So he's looking at heterogeneities across different bacterial cells. You can see some cells are producing more of the PHB relative to others, more being a whiter ratio color there. Okay. This is a really lovely example from Mark Hackett at Curtin University in Australia. So this was a mouse brain tissue. I've got an optical image of it. This is a great example of the multimodality. So we've got optical image here, we've got an autofluorescence image here, and now we've got O-PTIR images. So these are all taken from the same objective. Right? So we're not moving anything. So they're fully registered, fully overlayable. And then if we've taken some, so this image here actually is a 3-color RGB image where each of the colors actually is a ratio image in itself. So the nucleic acid, the red channel there, was a image generated from these two bands. So that ratio to that. The creatine was 1404 ratio to the protein band. And then we've got the lipid. So we've got 1738 ratio to 1655. And then each of those ratio images have been converted into an RGB overlay here. So you can see very clearly at a relatively coarse 2 micron pixel size, some of the lovely features we see there, and we can then zoom in and recollect at a much higher pixel density. Now I've got that 200 nanometer pixel step size. You can see individual cell nuclei lighting up there as well. Right, so it's also nice that the autofluorescence gives us a little added contrast, but in this case, it really is the beautiful label-free chemical images that I think are noteworthy here. That really kind of brings me to the end. I want to finish off so I can hand over to Christoph to talk about his exciting work. But I do want you to sort of take away from my half here that with O-PTIR, we literally are breaking any of these or all of these traditional IR and Raman limits. There's no more compromise or trade-offs between spatial resolution and sensitivity. You don't have to tolerate Raman fluorescence in order for good resolution, for good spatial resolution. You can now get the same Raman resolution with O-PTIR without any fluorescence interferences. It's highly water compatible, as we've seen as well. With O-PTIR, we've taken the traditional IR spatial resolutions of sort of 5, 10, 15 microns to submicron, even now sub 500 nanometers, all with excellent sensitivity. You don't have to go to synchrotrons anymore to try and get the best spatial resolution. You can do so in your lab. We can work in reflection mode, which is something that regular IR systems cannot do, and that makes measurements so much easier. The addition of co-located fluorescence, I think, has been a big boon to life science researchers. And again, Christoph will talk about how he's used fluorescence to help guide his measurements. And I've already spoken about that. And glass, of course. Glass, where you can use glass, it's wonderful because it's cheap, it's ubiquitous, and it really aids in the translation for O-PTIR into more traditional life science workflows. And with that, I'm going to hand over to Christoph. Christoph, please take it away. Thank you.
Yeah, thank you, Mustafa, for the introduction. And it's my pleasure to introduce you to our recent research, the fluorescence-guided submicron infrared spectroscopy of white blood cells in blood smears on glass slides. Yeah, the outline of my presentation is give a brief introduction into the topic of hematology. Then I refer to a recent publication of O-PTIR for leukemia cell differentiation. And third, we studied stained blood cells and calcium fluoride and compared O-PTIR with FTIR imaging. Then the next topic was unstained blood cells on glass. And yeah, we did a pre-screening by O-PTIR imaging. Then we prepared a subfraction, the so-called blood molecular. Sorry, I have to restart because stop here. I need my pre-screen
Yeah, sure.
window and so should I start over with the first?
If you
Yeah,
want.
I think I will
Okay.
start
Okay. That's sure. All right.
if I start from the beginning. Yeah.
Okay. No worries. All right.
Thank you, Mustafa, for the introduction. It's my pleasure to present our latest research on fluorescence-guided submicron infrared spectroscopy of white blood cells and blood smears on glass slides. I'm Christoph Krafft from Jena, Germany, and I'm working in the Leibniz Institute of Photonic Technology. Yeah, first I will give a brief introduction into the topic of hematology. Second, I will refer to a recent paper of O-PTIR for leukemia cell identification. And then I will start with a comparison of O-PTIR and FTIR imaging for stained blood cells on calcium fluoride. In the remaining topics, we studied blood cells on glass. First, we used the pre-screening by O-PTIR imaging. Then we studied a subfraction, the so-called peripheral blood molecular cells. And finally, we combined the O-PTIR with a pre-screening by fluorescence microscopy because we are particularly interested to determine and study white blood cells. And finally, I will give a conclusion and outlook. Yeah, hematology is a branch of medicine concerned with the study, diagnosis, treatment, and prevention of diseases related to the blood and blood-forming organs. Applications include the diagnosis of infections due to viruses, bacteria, and parasites such as malaria. Another application is to investigate blood disorders like anemia and leukemia. Main methods are complete blood count, and this can be performed very rapidly by a combination of forward and side scattering and electrical impedance. And the results of a complete blood count is shown here on the right. It is evident that the red blood cells are 3 orders of magnitude more abundant than the white blood cells. So these numbers are per liter. So that means there are in 1 milliliter around about 1 million white blood cells and 1 billion red blood cells. And then you get further information about the differential blood count. That means the differential fraction of neutrophils, lymphocytes, monocytes, eosinophil granulocytes, and eosinophilic and basophilic granulocytes. Flow cytometry can also be combined with fluorescence antibodies, and then, for example, a combination of the side scattering channel, which probes the size and the granularity of cells, can be combined here with the CD45 antigen staining, and this is the logarithmic scale. That means here the monocytes and the lymphocytes, which are of particular interest, can nicely be separated by these two channels. However, in 10 to 25%, the automated blood count by flow cytometry is not conclusive, and then this well-established staining followed by light microscopy protocol need to be performed by manual inspection of cells. And for this here, blood smear are prepared, and this was already pioneered by Paul Ehrlich in his dissertation 1870s. He won the Nobel Prize for his work in immunology in 1908. And so this is a typical microscopic image with these high abundant red blood cells, and then a few leukocytes. Here the granular structure of the cell nucleus is visible, and then here, typical for lymphocytes, the large nucleus. So I think it's time for more advanced technique, and well, we think optical photothermal infrared spectroscopy can be part of this. Yeah, I think I don't have to explain much about the role of vibrational microspectroscopy in biomedicine. The most frequent applied methods are infrared spectroscopy and Raman spectroscopy. They give to some extent complementary information. They are both label-free and non-invasive. They require minimal sample preparation, and you can get molecular fingerprints of biomedical samples such as breath, body liquid, cells, and tissue. And this was presented some time ago in a photothermal webinar, which is still available, like other interesting webinars to the life science topic and other topics. So we are in a lucky situation in Jena to have already two instruments. The Mirage R has been installed in 2000. Was the first instrument in the field with 785 nanometer probe laser. And then 1.5 years ago, second generation instrument, the Mirage LS, with two probe lasers, 532 nanometer, 785 nanometer, and a fluorescence channel has been installed in a different lab in Jena, where we can also perform experiments under safety level 2 conditions. Yeah, this work has been published 2023. So we collected leukemia-derived O-PTIR spectra of leukemia-derived cells in aqueous buffer, and the typical infrared spectrum is shown here with some bands of biomolecules, the amide-2 bands. The amide-1 band overlaps with the water band, and here's the ester band of lipids. And if a second derivative is calculated here from 4 different leukemia-derived cell lines, THP1, YOCAT, RAGI, and HL60, you can resolve more than 25 bands. That means here this board features, they contain substructures which become visible in the second derivative spectra. And if we look closely, in particular here in this amide band, there are variations between the cell lines, and these variation can be used for differentiation. O-PTIR can also be used for imaging, and so from some cells we collected images, and then at 1090 spectral contribution of nucleic acids can be probed. 1550, the amide-2 band probes protein distribution, and the band at 7045 was used to determine the lipid distribution. And the lipid channel is particularly interesting because there are cell-specific variation And yeah, in the future, we would also like to use this information to improve the cell differentiation. Now back to the topic of the main topic of the presentation, the blood smears. And yeah, in transmission, you see again here after staining red blood cells and the stained nuclei of the white blood cells. And if you collect, if you compare the spectra of white blood cell and red blood cell, there are certain differences. If you collect a different spectrum, you see elevated spectral contributions in white blood cells. However, these elevated spectral contribution in the different spectrum, they are due to the staining. And these staining molecules give quite intense signals in the O-PTIR spectrum, because the infrared laser and the probe laser is focused a very small spot, and you get a very pure spectrum of the stain of the staining molecules. The unstained red blood cell spectra are very similar between O-PTIR and FTIR. Yeah, if you compare the discrete wave number images, you see, well, as expected, that O-PTIR gives a much better lateral resolution. For example, here a single red blood cell can be resolved, and even fine structures can be resolved. And it was FTIR, the lateral resolution is in the range of 10 micrometer, and red blood cells cannot be resolved. You get a contrast at this 1604 wave number band, which is typical for stain. So then you get a good contrast for the white blood cells. Here in reflection, the staining is less evident than in transmission, but this is also a typical size of a white blood cell, 13 micrometer diameter. And in an RGB image, you can improve the contrast. The red blood cells are, well, in the red channel, and the white blood cells are then in the blue channel. However, for cell screening, the spectral overlap with the staining molecules is problematic, and therefore it's well, easier to use unstained cells, unstained blood smears, and also the use of glass slides makes, instead of calcium fluoride, makes this approach less expensive. So an overlay of a spectrum from red blood cells and white blood cells indicate several differences. The spectra were normalized here to the amide 1 band, and then here the red blood cell has a more intense amide 2 vibration, and the CH2 vibration in the high wave number range are more intense for white blood cells. Here in the low wave number range, there are spectral contributions from glass that are quite broad, and here the shaded areas are the standard deviation. And here it makes sense also to calculate the second derivative, because these broad spectral contributions of glass can be very nicely compensated in the second derivative. And then as another marker band to distinguish red blood cells from white blood cells. Here, the DNA band is resolved for white blood cells. There are further changes. For example, here the shoulder at the IMI2 band at 15-16 is evident, and then also a prominent feature is also for white blood cells, the shoulder in the IMI1 band, and as expected, also here is the symmetric, antisymmetric CH2 bands are also elevated in white blood cells. So we also collected images here. The total field of view was 1000 by 1000 micrometer. The step size 2 micrometer per pixel. So the speed was of the stage was 2000 micrometer per second. So discrete wave number image took approximately 500 seconds, and well, the contrast at CH2 band 2922, and here the amide 2 band at 1546 is too low to identify white blood cells. This is why a ratio was calculated. The ratio 2229 2922 divided by 1546, and then with the proper thresholding to avoid singularities. Here the white blood cells can be distinguished, and this is also confirmed here with the white light microscopy. But of course, in the unstained cells are really difficult to identify with a naked eye. And then after the white blood cells have been identified, 5 points were defined per cell, and from these 5 points, a spectrum was collected and averaged for further analysis. Yeah, well, identification of 10 white blood cells in more than 20 minutes was not very impressive, and therefore we try to improve the speed. And one option to improve the speed and the throughput was to remove granulocytes and red blood cells by a biochemical protocol, and the remaining cells, the monocytes and the lymphocytes, were then smeared also on the glass slide. And then we also, a starting point was to collect O-PTIR image. Here the field of view was slightly smaller, 500 by 500 micrometer. Took approximately 4 minutes, so much faster. And we also selected 2922 and 1546 as a wave number, and then several hundreds of PBMC were identified. So it was no problem to select cells for O-PTIR spectroscopy. Yeah, we collected here and we generate an RGB image to give a better correlation with the white light image. Yeah, so all together we collected 241 spectra, and this was required for the chemometric data processing, such as hierarchical cluster analysis. So but here a small complication was that the glass background was elevated, and after calculation of the second derivative, there were some fringes, so this spectral range was excluded from hierarchical cluster analysis. So we focus on the range from 1222 to 2992. Of course, in the central part there was a gap because the quantum cascade laser do not emit between 1800 and 2700 wave number. So, and then using hierarchical cluster analysis, two fractions of cells were identified. We called it PBMC1 and PBMC2. The smaller fraction are the monocytes, and the PBMC1, the larger fraction, was assigned to lymphocytes. And of course, it's well known that there are different types of lymphocytes, small and large lymphocytes, and T cells and B cells. So and these classes are responsible for the subdivision here observed in the dendrogram. Yeah, but of course it's better to screen blood smears without a biochemical preparation, and which is also time consuming. And so we found that in the fluorescence channel, in the blue and in the green fluorescence channel, the white blood cells and also here the platelets, these are the smaller particles evident, give an inherent fluorescence contrast. And the red blood cells give a weak contrast in the red channel. So, and you can generate in an RGB image this colorful image, but even in a single fluorescence image, which is a monochrome image, you can identify the white blood cells. And if you look carefully, you even see the granular structure of cell nucleus. And this is very quick, so a fluorescence image can be collected in less than a second, and this helps to select the white blood cells for infrared spectroscopy. And here also 5 points were defined per cell, and then a mean spectrum was calculated, and the mean spectra were subjected also to a hierarchical cluster analysis. And at a certain distance, we defined 4 clusters. The smallest clusters, 10% of the spectra, were assigned to monocytes. The green cluster were assigned to lymphocytes, and the other 2 clusters were assigned to granulocytes. And here these fraction of cells that we found, that 10% of the orange cluster coincide well with these automated flow cytometry blood count. The 37% agreed with the lymphocyte fraction, and the 53% with the neutrophilic granulocytes. Taking a look at the spectra, we found that the shoulder and the amide-1 band, and also the elevated CH2 bands in the second derivative are more intense in granulocytes than in monocytes, and this has been confirmed also in these in these PBMC, also these peripheral blood molecular cells. Here these cells also showed a small shoulder, and the reason is that the CH2 bands they are more abundant in the cytoplasm, and these granulocytes have a smaller nucleus and a larger cytoplasm. So this can also be biochemical be explained. Yeah, as a main conclusion, the fluorescence guidance is a method of choice for rapid pre-screening of white blood cells and blood smears on glass before then O-PTIR spectra can be collected. There's some further steps to improve the throughput. First, of course, the white blood cell selection based on fluorescence contrast can be automated. So maybe one field of view after the other can be screened, and then a spectrum is collected from each cell, one or multiple spectra. Of course, the assignments need to be validated by a gold standard, and the gold standard is still staining procedure. So we stain the cells after the experiments, and then there is also a microscopic inspection needed. And then if we confirmed a sufficient number of cells, then we will start to train supervised classification algorithm. Oops. And then the interesting work can start. Once we establish the workflow, we will start collecting data from patients with infections, leukemia, and other hematological disorders. And also of particular interest of, for example, question to determine the immune cell activation in white blood cells. Some experimental consideration. Yeah, we have to check if more than 5 spectra need to be collected per cell to better consider the variation in the cell. In the past, we used Raman spectroscopy for cell screening, and then we expanded the laser so that the laser focus collected the spectrum from each cell. Another option is collect an image with a wide field approach, wide field O-PTIR approach, and of course, if the QCL laser offers faster tuning, this could improve the throughput. You can select a spectral range of interest. This also would reduce the exposure time, or the fastest way would be to select discrete wave numbers. But yeah, this is a topic of future work. Acknowledgement, this work was done by Shravan Raghunathan, a PhD student in my lab, supported by other PhD students and postdocs. Our clinical partner, Dr. Michael Kienthoff and Dr. Susanne Piele, were from the Institute of Clinical Chemistry and Laboratory Diagnostics at the local university hospital. And the work was funded within the Leibniz Center for Photonic in Infection Research, which is funded by the Federal Ministry of Research and Education. And now I'm happy to answer questions. Thank you for your attention.
Thank you, Christoph, for that great presentation. I've got to say, when I saw this your word come out at the SPIE conference, I jumped on it right away. I thought this is a perfect demonstration of our vision as a company. I think I said this in my opening presentation as well. We wanted to introduce fluorescence as an added modality on this platform to help enable a more routine adoption of these sorts of spectroscopies into regular life science workflows. And you've done exactly what we envisaged, is that you're adding value to existing workflows with spectroscopy, perhaps where existing workflows have some gaps to them. And so you're bringing chemical information to those fluorescence images and helping guide the measurement. So really, really exciting work. I hope you continue along that line of research. Now, as you were presenting, I've seen a few questions come in. I think you've actually answered some of those towards the end already, so I won't read those out. One, I think, good one that I thought as well is why was the glass broad peak stronger in some of the cells and not in the other cells?
Yeah, that's a good question, and we discussed it also internally. And surprisingly, if you collect infrared spectrum from a pure glass light, the glass bands are extremely weak. And so only when you focus on the cell, the glass bands are elevated. So I suspect this could be a lensing effect, because for the small red blood cell, the glass background is less intense than for the larger white blood cells. And then another factor could be the blood smear was almost a monolayer. And here, when we studied the PBMC cells, so we had isolated cells, and the cells were also slightly larger. Here, the background was even more intense. Yeah, so it seems to correlate to some extent with the size of the cell. So, and therefore it's difficult to compensate the spectral contribution of glass by a glass spectrum, because it's difficult to collect a glass spectrum, and but yeah, in some cases calculating a second derivative is very successful.
Thank you for that, Christoph. And my additional comment on that might be that, in my experience with it, the sample thickness is also perhaps the biggest factor. So I suspect for the thinner cells, you will see definitely larger glass relative to protein peak. So I suspect the biggest factor there is going to be the thickness of your cells.
So, and we also studied in the past O-PTIR data from tissue sections on glass, and for this homogeneous tissue section, the glass background was fortunately also quite low.
Actually, I'm glad you mentioned that, because the amount of glass is going to depend very much on your thickness. So there is a certain thickness beyond which glass doesn't impact at all. So your typical histological sections that are, say, 7-10 microns, thereabouts and above, glass typically has zero contribution. And when you get lower than that, then the amount of glass becomes proportional to the protein band. So the thinner, the stronger the glass will be. A couple more questions. I'm also mindful of time. Somebody's asked, did you also try any simultaneous Raman on these cells?
Yeah, Raman was, first of all, we used the 785 probe laser. With 532, there were fluctuations in the spectra, because the red blood cells and also absorbs 532 nanometer radiation, and so 785 gave much better spectra, but the background was still too high to get reasonable Raman spectra even with 785 nanometer excitation.
Oh, great. In fact, that wasn't a loaded question. That was actually a great question, because I think it clearly demonstrates that there are certain cell types and certain tissue types as well that are very fluorescent, even with 785, as you just said. And that's where I think O-PTIR really does make a difference in that, with its zero fluorescence background. I think I do need to call it a day for this webinar, mindful of time again. So thank you, Christoph, first and foremost. It's been a while since we've done our first webinar. Hopefully we can do a lot more in the near future as well. Thank you to all of the attendees for dialing in and listening and taking time out of your day. All this is being recorded, so we will be sending out an automated email in the coming days with a link so that you can rewatch it or share it with your friends and colleagues. And with that, I will bid everyone a good day, farewell, and till our next webinar. Thank you. Bye-bye.